Animals live in an environment that's constantly changing. We need to have a way to track and assess these changes. We want to know, is there something new in the environment? Would it be risky to venture out and explore it? What's the potential reward of doing so? What are others in my social group doing? Responding to this information is a brain-wide effort. It's going to involve coordinating many different neurons in many different regions of the brain. The vertebrate nervous system accomplishes this using what we call the neuromodulatory systems of the brain. These are of great interest to neuroscientists because they have such a strong influence on behavior and cognition. Clinically, these are also very important because they play a role in many diseases, and they are the primary target of a lot of pharmaceutical interventions. Well, what do I mean by the neuromodulatory systems of the brain? I'm basically talking about the neurons that are going to release four transmitters here: norepinephrine, dopamine, serotonin, and acetylcholine. I should say that in addition to these four classic neurotransmitters that can modulate brain activity, there are a number of peptide neurotransmitters that also coordinate and regulate brain activity. We'll leave those for a different video. Neuroscientists group these neuromodulatory systems together because they share a number of common properties. First, each of these consists of relatively few neurons. There might be somewhere between 1,500 to 10,000 or so in a rodent, maybe between 20,000 and about 500,000 in humans. And those sound like pretty big numbers, but remember there are 75 million different neurons in a mouse. There are 86 billion different neurons in a human, so this is a tiny fraction, much less than 1/100% of all the neurons in the brain. The cell bodies of the neurons that produce these transmitters lie in the central core of the brain, so they'll lie in the brainstem or in deep cerebral nuclei. Now, although there are relatively few neurons, each neuron can influence many others. There might be more than 100,000 postsynaptic contacts per neuron, and this makes sense if you think about the fact that we have a handful of cells and we have to coordinate many different regions of the brain. Another property is that transmitter release can occur not only at synapses but also extrasynaptically. So there are swellings where these neurons can release transmitter onto glial cells or onto blood vessels or just into the extracellular space. So this allows the molecules to diffuse and again coordinate the actions of many different target cells. Finally, these transmitters are typically going to act through metabotropic receptors, so the actions can be long-lasting. They might last hours, and that's an appropriate timescale if we're thinking about sleep-wake cycles or mood. Now I'm going to briefly walk you through the four different neuromodulatory systems, beginning with noradrenergic neurons. In this system, the transmitter is norepinephrine. Norepinephrine is the preferred name for noradrenaline, which is an older name. It gets a little confusing in neuroscience because even though we call the transmitter norepinephrine, we talk about the neurons as being noradrenergic, and we'll talk about noradrenergic receptors. I think it's because it's easier to say noradrenergic than it would be to say norepinephrinergic. Noradrenaline is released from cells in the locus coeruleus. This is a nucleus in the pons, and actually I should say it's a paired nucleus because we have one on either side. This is really the smallest of the neuromodulatory nuclei. In humans, there are only about 12,000 neurons per side. Despite the small number of neurons, they will project to nearly every cortical area, so through the frontal, parietal, occipital, temporal cortices. They'll project to the thalamus and the hypothalamus, to the brainstem and to the spinal neurons. But I want to note here that although they project to the thalamus and hypothalamus, they don't project to basal ganglia. Norepinephrine's actions tend to be excitatory. Locus coeruleus neurons are implicated in arousal, attention, autonomic function. They're more active during wake than in sleep, and they're most active when a novel, non-threatening stimulus enters the environment. So if something new happens in the environment, it will elicit a burst of activity in the LC neurons. The second neuromodulatory system is the dopaminergic system. Here, the transmitter is dopamine. The cells that release dopamine lie in two clusters in the midbrain. These are called the substantia nigra, pars compacta, and the ventral tegmental area, and I'll illustrate them here. Now, the substantia nigra and the ventral tegmental area are adjacent to one another. In some sections, they even appear continuous. We treat them as two different nuclei, however, because they have distinct projections. The cells in the substantia nigra project to a region called the striatum. This is part of the basal ganglia. It includes the caudate nucleus and the putamen, and the function of this pathway is to facilitate the initiation of voluntary movement. Destruction of the dopaminergic neurons in the substantia nigra results in Parkinson's disease. The ventral tegmental area has two subsets of cells. One subset of them project to what's called the nucleus accumbens. This is called the mesolimbic projection, or sometimes the reward pathway, because the nucleus accumbens is associated with reward and with addiction. The other subset of cells in the ventral tegmental area projects to the cortex, especially to the prefrontal cortex, and input from these cells is involved in assigning value and reinforcing behaviors. The third neuromodulatory system is the serotonergic system. Here, the transmitter is serotonin. Serotonin is released from a chain of nuclei that spans from the midbrain down to the medulla. It's called the raphe nuclei. Raphe means ridge, and there's a series of these nuclei that lie in a ridge like a mountain chain on either side of the midline. Serotonergic neurons have extensive projections. The more rostral raphe nuclei project to the cortex, to the basal ganglia, the hippocampus, amygdala, thalamus, hypothalamus, and cerebrum. So you can see the extensions through the cortex, but also to regions like the hippocampus and the amygdala. This input seems to function in reward assessment and impulsivity and harm aversion, anxious states, mood, sleep, memory consolidation. The more caudal nuclei in the medulla project down to the spinal cord, where they function in modulating pain. The last neuromodulatory system is the cholinergic system. The transmitter here will be acetylcholine. The cells that release acetylcholine lie in two different sites in the brain. There's a cluster of nuclei in the forebrain. We call these the magnocellular cholinergic nuclei. So think about a series of several different nuclei clustered deep in the brain, covered by the cerebrum, but in the front part of the head. The different nuclei get names. The best known might be the medial septal nucleus and the nucleus basalis magnocellularis, which is also called the basal nucleus of Meinhardt. Now remember that a nucleus means a cluster of cell bodies, and I should say that not all of the cells in these nuclei are cholinergic, but there's a bunch of cells in each one of these nuclei that releases acetylcholine as a transmitter. The other place where we find the cell bodies of cholinergic neurons is in the brainstem. And again, these are given names like the pedunculopontine tegmental nucleus and the lateral dorsal pontine tegmentum. For our purposes, we just want to know that there's a cluster in the brainstem. These project to different regions. The forebrain complex projects to the cortex, to the limbic system, including the entorhinal cortex, the hippocampus, and the amygdala. It also projects to the olfactory bulb, although we can't see that in this image here. The brainstem nuclei project to the thalamus and the basal ganglia. I've gone through a lot of information quite quickly, so let me close by giving you a conceptual framework that may help as you think about the neuromodulatory systems. So here what we can see on the bottom row are the four different sets of neurons: the locus coeruleus releasing norepinephrine, the ventral tegmental and substantia nigra releasing dopamine, the raphe nuclei releasing serotonin, and the basal forebrain nuclei releasing acetylcholine. And these then send projections to a number of different regions in the brain: the cortex, the striatum, the hippocampus, and the thalamus. What I want you to see is that the different parts of the brain are getting input from multiple different neuromodulators. Now I will caution you that that doesn't mean that every single neuron is seeing each of these neurotransmitters. For example, in the cortex, which is a laminar structure, input from serotonergic neurons may go to a different layer than the input from dopaminergic neurons or the noradrenergic neurons. It gets even more complicated because there are interactions between the nuclei themselves. So for example, cells in the locus coeruleus send projections to the basal forebrain nuclei, and the raphe nucleus innervates the ventral tegmental area. And sometimes there are reciprocal innervations. So for example, the raphe nuclei and the basal forebrain innervate one another. Maintaining the balance between these different neurotransmitter systems is essential for normal brain function, and disruptions in this are linked to a whole host of diseases. Again, I've color-coded these here. And you can see, for example, that disruptions in the serotonergic input to the cortex could be linked to anxiety or ADHD or depression. Input from serotonergic neurons to the cortex could also be linked to ADHD, but also to OCD and schizophrenia. Disruptions in the locus coeruleus neurons could lead to stress. Disruptions to input to other brain areas, like the hippocampus, can be involved in memory deficits and in depression. At the start of the video, I said that these neuromodulatory systems were a primary target of pharmaceuticals and in how we treat these different disorders. I hope you can see, though, that because of the interactions between these different neuromodulatory systems, simply treating one of them may not be sufficient to alleviate all of the symptoms. Given the prevalence of neurological disorders such as anxiety and depression and ADHD, you will not be surprised to learn that this is an area where there's a lot of active research.